Okay, I think this is the time you've all been waiting for, your chance to ask your questions. Some of the people you have met already this morning or this afternoon, and just to add, and they're also in your program, Nikolai Narishkin, PhD from PTC, and also Ed O'Mara from PTC. And I think you probably recognize everybody else. So do you all want to say a few words first, or you want to just start out with questions?
Okay, so my role is to moderate this meeting. If you have bad things to say, you tell them to me. If you have good things to say, you tell them to the panel. So if there's a burning question, we start. If not, I can slowly start. Let me put people on the spot, right? Let's start with Dr. O'Mara, right? Whom you haven't met, and I'm purposely putting him on the spot. It's the first time that he's at FD Day, and he works with PTC. He's of course a physician by training, but he has also been involved in drug development for a long time, and relatively recently he has been assigned to the FD space. So do you mind if I call you Ed, Dr. O'Mara? Ed,
please. Yes. Go ahead.
What's your first impression about the FD community, or FD as a rare disease, particularly if you compare it with other orphan diseases with whom you've been working?
Okay. Well, again, just to actually jump back for a second, because
Can you use the microphone?
is that better?
Yeah.
Okay.
It's voice activated. So
Oh, okay. My experience in the past, and I've been in industry for 20 some odd years, has originally started as large disease-based diseases that are really indifferent to the patient population. You know, people have the disease, you put them in clinical trials, and you get a result. Here, I've entered in working with PTC the small disease space, which is such as familial dysautonomia with only several hundred people. Every person matters. Identifying everyone who can participate in these programs really is important. It is a much more personal interaction at all levels of how we develop drug. My responsibility will be as the physician who first sees patients in the clinical trials. Therefore, I have a special responsibility to you all to lay out very clearly why what we're doing is very important, so you understand fully the benefits, and as well as when you do clinical trials, the risks attached to what we propose to do. One other very important thing that has changed more globally in the last number of years, and I would say, dare say in the last 5 years, there's been increased attention from regulatory authorities, particularly the FDA, that there's a large number of diseases that are not being attended to by industry or any organization that produces therapeutic results, produces drugs for use. So something had to change. The paradigm in which the regulators operated had to change to be much more flexible. And as one thing that Dr. Kaufman, Lucy Kaufman in particular, had pointed to is that natural history databases, because the population of people with the disease is so small, you cannot afford to use patients in placebo arms. You have to engage every single one as many as you can use. The understanding here is that the FDA will be more flexible with us at every step of the way. They will be more involved with us. So the relationship that you heard about earlier of academia and the pharmaceutical industry, as well as patients and their advocates, is really now expanded to include regulatory authorities, because none of the groups cannot be involved from the get-go and has to have a say in how things develop. You don't want to take all your hard-earned efforts in the form of clinical trials and bring them to the FDA or any regulatory organization for that matter late in the game. They may have objections, they may have concerns. We will, as we develop PTC 258, be very involved with the FDA, and they will be involved with both us and through us with you folks to help design the database to make it optimally useful, to make sure that the measures that are important to you get incorporated in the way we design and execute clinical trials. So this is a great time to be involved in rare disease. Why? Because everybody's listening. With that in mind, and it looks like everybody here is laser focused on this disease. I look at your faces and they're very, very clear. We will do our best to keep you informed as we can, as we develop drugs, to give you a sense of what we do and, to the best of our ability, why we do things. We will continue to work very closely with our preclinical colleagues, those people who are working diligently in the labs, to provide a basis for us to make the leap from cell-based or animal-based discoveries to those that are translatable, if you will, or relevant to all the patients out there. And so with that said, hopefully I've answered your question and not gone on too long. I'm happy for the next question, if that helps.
So thanks so much. And let me maybe continuing with the thing, and I'm putting the PTC folks on the spot. Maybe something that doesn't have an easy answer, but Nikolai, we are all aware that there are close to 8,000 rare diseases, right? How did we get so lucky? How did we get PTC's attention to the one that we are focused on, and how did we get PTC to pick the challenge?
Well, it really it was a combination of several factors, and this is really a testament, first of all, to the work that Sue's lab has done over the years, to the work that the Autonomia Center at NYU has done over the years, setting the stage in both understanding the disease and the molecular basis of the disease. That's very important because we are a precision medicine-based company. We are focused on targets that are well defined. Well, actually, it helps everybody, but we are particularly, in our 21 years of drug discovery and development experience, have focused on diseases that are related to gene expression and have very specific genetic basis. So the work from Sue's lab really made FD such a disease with very well-defined genetics and very well understood mechanism of the pathology. And then the natural history, or more generally, understanding what the disease is and how it evolves over time. That's the work from Felicia, from Horatio, and the team here at the center. That was the second big component that really helped us, put us on our radar screen and sort of made us very aware of it. The beauty of the internet age is that literature and sort of the advances are very rapidly disseminated, and we keep an active look on all of them, and this really very quickly came to the top for the reasons I described. But then the third component was really the sort of the openness and the willingness of all the people I mentioned to collaborate and really to be together as one team. We don't think about this program as PTC program. It really is not. It's a joint effort. We're here together with the center, with the labs. It's not now just Sue's lab. It's also Francis's lab, and we're looking to expand because there are additional labs whose work is becoming important, and we are engaging with. So Sue's slide mentioned a couple of additional activities that are starting, and then of course, so the clinical center, the foundation, the academia, and then our colleagues, a large group at PTC. We're on this together. And it was really, it was not a coincidence. It was the reasons that I described that really made it possible. And we're very grateful for the work that had been done before we joined, and we're looking forward to continue together.
So thanks so much on that. I'm happy to keep asking them to elaborate on things, but also you should know that any questions you have, yes, go ahead, from Canada.
From Canada. As a parent, yes, two questions. One, you talked about PCT, the lab working on exploring the retina involvement and the decrease in neurons and so on. As a precursor, there was Dr. Mendoza, am I saying it right? who had talked about whatever research he was doing or the data, and was like imminently, going to have some effect on more neuron development. Is Dr. Mendoza, are you working with previous clinicians?
Yes, yes. Not only we are working, he's also, Dr. Mendoza is at Bascom Palmer now, Carlos Mendoza in Miami. He had to go there for a few years due to a licensing issue, had to be in Florida. So, but he's still heavily involved and participates looks at the OCTs and is part of the team and has spoke. PTC, it's not PCT, it's PTC, but PC is the Communist Party in South America. This is the PTC. So this is the Workers' Party. It's a different party. In Brazil, it's called PT, the Workers' Party. Now, the issue is that Dr. Mendoza is still involved with PTC and participates in the advisory meetings. Fortunately, there are two more people in ophthalmology. One is Quintus, and I can never pronounce his last name, who's a visual scientist from PTC, who's also working on that, and then Lila Raju that you met this morning. But Carlos, as a retina person, is also heavily involved. That's what you want to know.
That's what I wanted to know.
One thing I will just add about ophthalmology is that they love to split things into the front of the eye and the back of the eye, right? So you have, like, Carlos looks at the back of the eye and Dr. Raju looks at the front of the eye. Right, so we're putting all that together, and now we're fortunate that Bar just joined our team, who is really going to help us like centralize that data and understand it much better. So we need both. We need Carlos plus other specialists in the eye. So it's great we continue to have him involved, and we also get other people into the forum too.
I asked because
Yes.
this morning was about corneal abrasions, but then the other part is the retina, and I guess where's that research and where's it going?
They always divide front of the eye, back of the eye. It's
So
small.
the point, Lucy is absolutely right. The point is that for the front of the eye, the cornea, there are a lot of things to do in terms of preventing problems, right? The cornea is like the lens of the camera. The retina is like the film. The retina is the one that gets progressively worse because of the nerves dying, and so far there's little we can do, although we think we will be able to do something perhaps with this new medication. For the cornea, for the front part of the eye, there are constantly things to do, and that's where the problems arise, the ulcers, right? But again, as Lucy mentioned, there are two different groups, right? One of the things, and perhaps Sue can elaborate on this, one, as he told you in her presentation, one of the limitations we had was that the mice model, was good to understand the genetics, but not the symptoms. Now that is changing, and with the new mouse model, there is problems in the eyes and problem in the spine, two typical features, two typical symptoms of FD, and now we believe that this will be good models in which we test the treatments, and probably Sue can elaborate on that.
I could say just a few words about the research going on for the eyes. It's further off than the development. I mean, we are hopeful that if we get a drug that is beneficial, that maybe that could help the eyes. But we still are working on understanding both the retinal defects in the mice. And so in Frances's lab, I'll speak on her behalf since she's not here, she's using something called gene therapy. She's going to test gene therapy, which you've likely all heard of, which is basically putting back a good copy of the gene into the eye. And so that's some research that's going on in her lab. In our lab, Elisabetta wrote a grant with Frances in order to create a new model, a new mouse model to study the eye, where we are just going to remove the gene from the eye so we can have a better mouse model to study. So that work's going on. And then we have another project going on in the lab using yet another way of delivering another kind of corrector, if you will, to correct splicing in the eye. So there's a lot of different research going on. It's not as far along as the development of our drug, but there is interest. The thing about that we have to remember, again, as I said, mice aren't humans. The mouse retina is different than the human retina, so we don't see exactly the same patterns of disease in the retinas, but we are looking to see exactly what the retinas look like in our mice now. So that is a newer line of work. So.
And as far as the timeline, and Nikolai, I appreciate the conversation that we had earlier, is that when it comes to the development of the drug and then testing results on our children, what is the timeline we're looking at, and what are we looking to see as we move forward over the next 6 months to a year or several years?
This doesn't specifically refer to dates, but I can walk you through in a general sense how it is not like traditional drug development. And where we are now is we're in the toxicologic or preclinical stage, and that usually takes about a year. Once you get that information, you submit that as part of a package to the FDA. You have to be granted the ability to go into testing people. And on that basis, the FDA has to approve that whatever preclinical testing was done was adequate to give you at least a general sense of safety and also some sense of potential beneficial effects. So you have those two features to go into the clinic. Where we see this evolving thereafter is you then go into the clinic, and we would look first to test the drug in healthy subjects. The purpose being healthy subjects, you can do the studies relatively quickly, there's relatively low risk to the individuals, and also we want to preserve those patients who could potentially benefit from the use of the drug from receiving doses of drug as we evaluate in an escalating manner from subtherapeutic or supertherapeutic doses. We want to hit it pretty much right by the time we get to the patients. And that usually evolves over about a year and a half. That information comes back, and what do you learn from that? You learn basic general safety. And you also learn, as we take blood samples from from healthy subjects, we learn the pharmacokinetics, how the drug gets in, how much actually distributes in the body, and then how it's eliminated. So we have a general sense of how it behaves relative to not only the effects of absorption in the gut, but also how it affects potentially the kidney and liver, as well as what metabolites you have to worry about. Because sometimes you have a perfectly safe drug that once it forms metabolites, they're problematic. And we want to make sure we've characterized that before getting anywhere near a patient. What makes it unusual here, and after that time frame, or maybe somewhat concurrent, we'll see how it goes, we'd like to see, can we show a reasonably useful beneficial effect? And so where we want to start with that is the carriers. We want to understand the carriers who are phenotypically normal, meaning they don't have any of the manifestations. But what they do have is the ELP1 level is lower. And if potentially we can raise that level, and we wouldn't have to do it in as extensive a study as we did in healthy subjects, we'd get a sense, well, gee, is our drug hitting the target and is it having a desired effect, and maybe what is the magnitude? Because what is necessary here is to get the ELP level to a reasonable level. It doesn't have to go back to 100%, but it has to go to some percentage that's far above where most patients reside, which is 10 or 15% of normal production in the neurons. So we're not quite sure yet whether 30% will do it, or 50%, or 70%, but at least we'll see that we can move the needle. Once we've gotten all that information, we look again at the safety, we look again at, well, how much drug is needed to produce the effect in the carriers. After that year or so, additional year, then it's on to the patients, where we have a tighter drug range, so we know how the drug would likely behave. It may be complicated to some degree, as there are GI disturbances, and how a drug gets in and is absorbed may be a function of how functional or dysfunctional the GI tract is. We're going to learn that as we go. And then we'll also be measuring ELP1 levels as well. And those also will go on to look at levels of, other markers which are in development, but also those are more phenotypic, how a gait is affected, evidence of kyphosis and so forth. And what we target in that population will be really driven by what we've observed, and we've done some statistical analyses in the natural history population. Because as Lucy brought up earlier, these different manifestations hit at a different stage of disease, different point of life, in a variable subpopulations of your very small group to begin with. So you're looking at a window. I mean, if you do the math here, it's probably in a 3 to 4 year window before we're really into getting the patients themselves, into the picture where we can understand how it affects them. And in that interim, we're going to continue to build a natural history database and figure out those features that will be most useful to add for both short-term demonstration effects and longer-term. Is that reasonable for you?
I guess I follow up.
Please.
So the first patients, the healthy patients, who are they? Are they people who sign up for a drug trial?
Yes. Yes, they would be unrelated to FD per se.
And then the carriers would look to us.
That is correct.
The parents.
That means you.
Siblings?
If they are carriers?
Is there carriers?
Right, well, yes.
Yeah.
Could be, yeah.
Yeah.
One of the initiatives we have in the clinic right now is not to only just collect samples of blood to measure in patients, but also carriers, right? So the whole reasoning behind that is that we believe the carriers are going to be really important for helping get these approvals faster in the earlier stages of development. So when, the kids are always laughing that their parents have to give blood, but, it's for a good cause, right?
My understanding was that there would be trials early 2020, end of 2019. And Lucy, when you spoke, you said, I think, if I'm quoting correctly, we don't have 5 years for trials and stuff, but you're saying this process is a 4-year process before our patients, our kids start to get any benefit. So I'm confused.
No, understood. I mean, there's a nuance here, and the fact is we want to get into the clinic as quickly as possible. There is another party that is very much involved in this, as I said, is the FDA. We have to work with them to negotiate the timelines, to negotiate how quickly we can progress. What I penciled out for the prior question was just a general sense of how we want to move. We can potentially move faster. It all depends on factors being favorable. It depends on how many people we ultimately enroll. And again, what we're trying to do is, because we have one opportunity with the patient population, understand that we want to make sure that we have it as well mapped out and characterized, because there are, as I said, there are risks attached to any therapy you introduce. To characterize those risks as best as possible before we introduce this to people for whom it really matters. And that's what we're trying to do. We don't want to expend, for the sake of saying, well, we're treating patients, well, no, not really. We have to establish the zone, if you will, where it's most likely to produce benefit for them, and not waste their time or their hope on agents that are not thoroughly characterized to the best ability before getting to them, because they have one opportunity to be right. And that's why I say yes, we're getting in the clinic. There's certain prerequisites to getting everything completely right, and everything, of course, is contingent upon what the FDA believes.
Right, so that's my follow-up.
Yes.
So two things. Time is of the essence for this
Yes.
population. I think everybody's quite aware of that. But my understanding also was, as what we can do, right, is that the FDA, and I could be wrong, I was told at one point that, and I'm assuming that the foundation and the center, unless the foundation will get involved, that the parents can apply pressure to our, politically, and write letters and make sure that people that are, involved, so that the FDA, so that we can make sure to get things approved. I don't know if it was when we went out and met, maybe you were with me, there was discussion about what we could do to help that process. And I don't know if that's still something that...
Well, I can't speak to that aspect per se, because that's a bit outside my scope. But one thing that I think would be very helpful is where the FDA has also asked for patient populations and advocates support is in the area of, well, what quality of life measures matter. And so surveys will be useful. Your ability to participate, giving them data to work with is helpful. It's as helpful as, say, having a political side to matter. Everything you can contribute with, in interacting with us as well, to get the scientific basis for what really shows therapeutic benefit will be very powerful tools to hand to the FDA for their agreement.
So maybe to add,
Yeah,
a little bit to add. I think there's also a component of disease awareness and sharing the stories of FD families and FD patients with the FDA, and just educating them and making them aware that this disease exists, and this is what it is. This is what a day in life of a family is, and this is what we need. We need to have addressed that it goes along the lines of how the disease progresses, but also making them aware and appreciate that there is high unmet need, that there is work ongoing to develop new therapeutic approaches, and a commitment from both the industry, the academia, and the patient community to move as fast as possible. So once they hear that message, and there are different constituents within the FDA that are tasked or will be interested in hearing this. Starting with the Office of Rare Diseases and Orphan Drugs, and then the specific therapeutic areas that may be assigned to review the application. And it will be very helpful and very actually important. So this is on the side of what the parents and families can do in the foundation. This is definitely on the list.
So I want to emphasize now what Nikolai said, and I go to Dr. Eisenson in a second. I was in two drug approvals for the FDA and presenting the data. I can tell you that one of the most powerful parts after the science presentation is the patients and the advocacy groups. In fact, in both cases, the approval was arguable because these are rare diseases and the trials were only a few hundred patients. And what really killed the balance for approval was the dramatic testimonials of the patients that makes the advisory members swing to approval. So you will have, Lori, a crucial, and all parents will have a very important role going to the FDA and talking about the issues. That's one. The other clarification is that Lucy, the point that Lucy made, is that yes, we don't have 5 years. She was referring, though, of course we don't have 5 years now, but she was referring to the actual trial. Imagine if we don't have good natural history data, in addition to waiting to start, we are obliged to get half the patients on a sugar pill for another 5 years. Well, that is what we cannot do. So that's why it's crucial to have data to compare and not have to enroll, even if it takes us a little later to get into clinical trials, not to have to include a placebo arm. Then, of course, I want to echo look, we are driving PTC nuts in saying, when are we starting? We are really in a hurry. We also have to understand, and they have a very good point, number one, that you cannot start until you have all the toxicology and all the pharmacology. That's number one. And the other part is that they do need from us a good outcome measure. They need something that they can measure that in a year or two can tell the maximum amount of time that the drug is working. And we thought we had many of those answers. They are very hard-nosed to convince them that these are good parameters, and maybe they are not that good because fortunately the disease does not worsen so rapidly, and whatever changes slowly and in many different parameters. So the reason we're focusing on the retina is not because the retina, of course the retina is important, but the retina is a part of the brain that we can look, take a picture and measure, and there is clearly something changing there. If you look in the teeth, if we will find that the teeth change a small amount every 6 months, we'll be golden also. What we are looking is at things that change and change quickly and that we can modify. So that's the big, in one sentence, that's the holy grail. If we, in other diseases that they are used to, like muscular dystrophy or spinal muscular atrophy, things get worse quickly and they get worse in terms of the strength. So that's relatively easy to measure. It's difficult, but it's difficult. In FD, what are we going to measure? Crisis, pulmonary infections, renal function, blood pressure. I mean, all these things are so multi-parametric that it's not easy to give a drug, even if it's magic. Even if we replace the gene, things are going to change in 6 months or in 3 months, and they can be, people break their bones, Not because they don't have neurons. They break the bones because they don't have feelings. We're not going to fix the bone, right? Or maybe we will. There are so many questions here that it's also understandable that PTC says, guys, let me get very, very candid. We are investing millions and millions of dollars. We have one chance to do this. We better do it right. That's one side of the coin. The other side is also, first, do no harm. I mean, what if this drug is fantastic and after a month we realize that it's destroying the kidney or destroying the liver or doing other things we didn't find? How happy are you going to be, right? You're going to tell us, guys, how you didn't find this out? So all these things we're doing, trust us, they are in a hurry and we are in a hurry at the same time. Like Napoleon used to say, dress me very slowly because I'm in a hurry, right? Meaning you cannot correct mistakes once you made them. You have to prevent them and know what you're doing before. So Dr. Eisenson wanted to,
Yeah, well, I think you read my mind, because you made it abundantly clear that studying kyphosis, studying retina degeneration, it takes too long and it won't give us definitive answers. Do we have any objective markers that we can use? Have you studied protein quantities in healthy people, in carriers, in patients? Is there something that you can absolutely delineate and use to determine efficacy of these drugs?
well, you'd also read my, yes, we are measuring. Let me give you some examples. Right, for many years, and Sue can talk about that, because Sue is a molecular biology lab and is a geneticist. What we were measuring was the messenger RNA, the signal, whether it was the wrong one or the right one. But we were not measuring the protein. The protein itself requires a different method to measure it, radioimmunoassay, and none of the methods were good. And we didn't really need that. We really, what Sue was doing with initial kinetic and what we all work on was in a drug that corrects the message, right? So the message, we were measuring the message also, not the final product, and that's just a laboratory trick. Now, with PTC help, and that requires deep pockets, a lot of resources and a lot of money, they develop a better radioimmunoassay for the protein, and now we can measure the protein. That's why we want everybody to give blood. We assume from initial studies that the level of protein in the carriers is a little lower. Many of these things are assumptions. We don't know them for a fact. We also know we have the measurements of all the population, but you know, many of these measurements and protein or messenger RNA were done anonymously. You could not legally connect the person. That person, you only knew he was a patient with FD. Now we need to know who's that person to connect whether the severity has to do with those measurements. Some of those things are not there because legally could not be joined. So there are so many, even bureaucratic issues, legal issues, that all those things have to be followed. You know, let me give you a silly example. For patients from Canada that may have some cognitive issue, some of the parents decide that they are not going to be their legal guardians because they say, why do we need to be a legal guardian? This person lives with us. So when they come here to participate in a study, we say, well, the person is 25, he's not a child, he cannot sign as a child. On the other hand, we are writing that cognitively is impaired. If somebody has a problem with cognition, the guardian has to, they are legally not allowed, right? So, but there's no legal guardian because the parent didn't do the thing, right? So we said, okay, what should we do? Let's have both of them sign, the child and the father. Well, bottom line, the IRB says, you didn't do one mistake, you made two. This person cannot sign and the other one cannot sign. So this is a federal problem. You made a federal violation. Your trial has to stop and you cannot continue enrolling people that cannot get consent. That just gives you an example. We tell them, guys, you are idiots. You don't understand anything and I'm sick and tired of all you. Unfortunately, you cannot do that many times. You do it once, you do it twice, you do it 3 times, they tell you you are hostile. Yes, I am hostile when they don't let me do what I want to do. Now, I want to tell you that there are many of these things that we have to follow. Those are examples, I give you examples that appear to be silly, but there are a lot of regulations, and PTC not only has to follow those regulations, then we have to go to the FDA that makes the IRS look bland. Right? So, and they have a mission. They have to protect people. Right? So when you go to when you go to the FDA, the image they have is that these guys they just want money. And we are all corrupt and we want to get fees to get this approved. That's the way you are received. You are criminals here that try to sell poisons that people are going to pay. That's how it starts. So we do have to tell them, guys, nobody's going to make real money here. There are 300 patients. You tell me what sort of money they're going to make. Still, I'm telling you these things that sound crude to tell you what real life is in terms of drug development. Yes?
So I'm going to go back to what we were saying before then. Does it make sense to then start early in the process with families to be able to talk about our stories and to be able to share? Because when that comes down to play, then if they're looking at stories that will push the needle there, then I assume that there's obviously a room here of people who are...
Sure, but they will ask you directly, and I'm asking you, and they will do the same when the time arrives, and there are these open meetings at the FDA. When you go to, first you present the data, the result of your trial. That goes to 15 guys of an advisory board, depending on which section, and then they invite people from outside. Then the people that did the trial go and present, like we are presenting now, all the results. These advisory people are very hard-nosed, and they say, well, you're only showing 0.3 here, you're showing, how do we assure of this, that? So they can say, go back and do more studies. That is when the patient's parents and the patients themselves have a role, and they say, guys, you're all scientists, it's very easy for you to be there questioning the statistics. We are the ones that suffer, we don't have another option, and surprisingly, that sways people. And many times you just need, the vote is, let's say, 7-5, or 8-4, or 6-6. You need to sway 2 or 3 people, and you get the drug approved. And if you get the drug approved, that makes a whole difference, because now they have a source of income for that drug and they can continue. So you will be called, trust me, you will be called to participate in that.
So I think just to go back and add something to both of your points, I think there's 3 really fundamental things that parents can do right now. Number 1 is get the story out. The more noise we make about FD, the more we tell how much people suffer, we have to get people listening to that story, and the foundation does a good job of messaging that, but the more we can do it, the more you can share your story, the better. The other thing, which goes back to your point of what are we doing for shorter term biomarkers, is please give blood. When you come into the office, we're asking you to give those samples of blood, not just the kids themselves with FD or the adults, but also the parents, because that is so valuable for us as a potential outcome measure. And the third thing is, we know that the kids having to go on the testing that we do for clinical care, they're not the happiest with that. But, being like you all do, like helping them understand that that's helping, letting us work quickly, but get the information we need. And we're always a little bit tentative when we ask the kids to give blood in the office, right? Because it's unpleasant, they don't like it, it upsets them, etc., etc. We're asking it because right now we really need it, right? So be on our side with that, and as you do, just help us get in there quickly and get what we need, right?
So maybe to add just a couple of sentences, I think you're absolutely right that the question before Larry's one was a good one, because there is a defined regulatory mechanism to get a drug approved, a subpart H or accelerated approval based on surrogate marker that is reasonably likely to predict clinical benefit. So it is an existing mechanism, and indeed what Lucy just described is other components that need to go into it. So we need these data. And we need to understand what actually is or can be the surrogate marker to reasonably likely predict clinical benefit. And this is an active research that we're all doing here, and this is part of the, you may have heard several times today already, the plea to participate in a natural history study as well as in the biomarker work.
You know, the idea, and goes to the question what Nikolai is also saying, would be to have an outcome measure that perhaps will be a composite. So if you have, let's say, the protein, the abnormal protein goes up 20%, the decay of the retina is X percentage. If you can put several things together and do a score, that's how this drug for TTR amyloidosis just got approved, and other drugs that PTC is also working on got approved, which are composite outcome measures. So, Mrs. Newman, yes.
I have two questions. Regarding the need for more blood from the carriers and more blood from the IPDs, is there a specific outreach going on to families who don't come to the center regularly, perhaps have not come to the center in numerous years? I mean, a lot of the families who are here today, and some of you on the panel may not realize this, like, we're the devoted ones. Like,
Yeah.
We're the ones who come to FD Day, and we're the ones who go to the annual appointments. There are those families, for various reasons, who perhaps don't. And some of them live quite close to New York. And I was wondering, that's my first question, you know, if there's been a specific outreach to try to get them to come in, if for nothing else other than to give you some blood.
Right. So let me take that one on. Lainey has been an incredible addition to the team because she is working on the basis that the natural history study is not about one center, one foundation. It's about all of us, right? She has met with other organizations with the goal of outreach and trying to reach those people. We recently had the protocol amended so that people don't have to visit us and be part of the study, which is the first step of just telling them, even if you do not come, you can still be a part of this. And then we have to figure out the logistics on top of that of how we would get blood samples from those people, which, you know, for us is a headache to try and solve. But the messaging around it is there. We are reaching out to the other organizations. We're working with the Israeli FD Foundation. We're working with FD UK and other organizations to try and get everybody behind this natural history study and this effort to get samples. But, you know, it's not about, you know, this project is not about the dedicated families that are here. Of course, you guys are important, but the rest of the population out there, we hope are going to see the importance of this, because it's about treatment and it's about getting it there. And hopefully we can spread the message that helping in this way helps the entire population, right?
So, and Lisa, you bring a very important point, and we would love if you suggest, and so will Lenny, more ways to attract people. I mean, we try. Look, with the Hasidic community, we bend over backwards to tell them, look, if you want your child can come in a day that there's nobody, right? They want to make certain that nobody else is going to be there. I mean, we don't offer Shabbat because they would not come on Shabbat. Because if not, we would tell them, we'll see you on a Saturday, right? So we try to make certain that they participate, and we want everybody to participate, to tell them. And if you have other ways, so you can think or propose or suggest other ways to bring more afflicted patients, we are all for it. In fact, I think we should do huge population screenings in Mexico. So then I think we will get a lot of patients, right?
Just another thing to add to that as well. If any of you in the audience or people that receive this message are part of another organization that works with FD, spread the message, right? I mean, you can be part of different advocacy groups, right? But if you are part of another advocacy group, you know, speak about the natural history study, because the more of us that speak about this, the more chance we have of bringing the other patient population in.
And they don't have to, let's insist on something. To participate, to give the information, they don't have to be our patients. If they, right? I mean, It's the same. We spoke even with Paris Rubin, saying, can we share data? The answer is, I don't need any trials, I just need belief. Well, we need belief also, but we also want trials. Yes.
So I have just a follow-up question. I'm wondering if shark thought plays a role in any of what's going on now. A lot of people have been giving our kids shark thought for a while now, because originally there was this idea, that...
Look, the idea is still there. It may be, it was not well reproduced. We were not able to reproduce that very well. We are not sure. On the other hand, talking also with PTC or with the scientists there, we assume it doesn't change things. We almost consider that as standard treatment because a lot of people take it. I tell you that the idea is a very good idea. It's the only compound that is not, the results are not being reproduced. I'm not putting it down. We don't know. We are not sure. It's very difficult to measure the protein. At the same time, it will not stop, meaning people that took phosphatidylserine can still be part of this trial.
Yeah. So I'm on social media, website, and emails, and I haven't heard about the history study. Is there anything, to help to spread the word, but I feel silly making my own posts? Is there anything out there?
Absolutely,
well,
absolutely.
We give you anything, we give you pictures, slides, descriptions, literature. If you tell us, we'll be more than happy.
We have the blog. Sign up for the blog, right? This is why this information, everything that happens in the treatment center, everything that's published, every important thing you should be part of is written there, right? That's the channel that we use to communicate with you, right? So www.dysautonomiacenter.com, sign up as an email follower, and you'll get every single post we have that includes detailed information for patients, right? So it's all there. Yeah. Yep, ways to share it. You can repost it on Facebook, on Twitter, on anything. Yeah.
And the more you share it and do outreach, will help and will love you.
Let me send it out too.
Right?
Yes, she does.
Perfect. That's great.
My takeaway on all this is that, different people have different ways of doing things. Some people spend their whole time on social media, others don't. I think a very simple, not complicated, very simple email from either the center, the foundation, to the population, saying that, talking about the need for blood tests and talking about at the right time the need to get something together for the FDA in a mass form, that's the way to do it. And whether it's through the blog or through a simple email, I don't look at the blog, maybe. I happen to be in New York this week. I'm happy to come in for a blood test this week if that's helpful.
We never heard about it.
I mean, it just has to be very simple email to the whole population.
You have a good point. I can disclose. I don't use social media either. In the center, they tell me you're the only person that uses the fax. I still use the fax, and I still think that was a great invention. It changed my life. Right? Now, I don't use Facebook, Twitter, just the fax. So, and I guess you are the same. Now, email, when I talk about email, I get the same responses like with the fax. People say, email, nobody reads emails. Okay. So, but we take your point. I'm just sort of joking that different people get we'll do our best to send the message out even more.
Yeah.
But just to confirm that we have done email blasting in the past about the natural history, so we have used that method. So maybe it went to your spam folder, right?
No,
but we've done that before.
We start with the fax.
You have to keep it even more simple than that.
Right.
Just say, we need you to come in for a blood test, and then you can explain why.
Perfect.
Keep it very simple.
Marisa will speak with you about donating blood next week, right?
There is Marisa. Look, she got all the survivors from 9/11. She should be able. I told her you should be able to get the FDs.
Can I throw in a pitch for Horacio?
Yes.
If you come in to give a blood sample, please give a poop. We call we call it brown gold in my lab. We can discover all kinds of things in your GI tract, but we can't do it without that sample.
Well, it's great that Seth, I'm going to switch a little bit, it's great that Seth is here. We should also take advantage of him. You know, this may be a seismic change in understanding of a number of things. You heard his talk, right? I mean, he talked about the 100 million neurons in the gut, but he doesn't tell you that there are billions of bacteria, right? So there are so many bacteria in the gut, and the connection between the bacteria and those neurons, this may be really a game changer. Of course, everybody asked Seth and Francis whether they should take probiotics. So they hate that question, but I'm going to make it because instead of you asking it, I'm going to take Seth, and they hate that. Do you think they should take probiotics? Well, because probiotics, I heard on the radio, have all these good bacteria. Should I get probiotics for my child?
Well, it's interesting this comes up in the context of FDA, which doesn't not regulate probiotics because they are lobbied by pharma to produce the product, but not have to show the results. What I can say is probiotics are probably not going to hurt anyone, right? There's been a lot of folks that take probiotics and either don't feel an effect or feel an effect and the effect goes away. So by that standard, it's probably okay. What I can't tell you is that it's going to help. And from a microbiology perspective, the probiotic that you get over the counter probably won't establish in your gut. And for that aspect, I really don't have high hopes until we can look at individuals and personalize those probiotics. So this is why we need that information to help figure that out.
So Seth, that's the easy answer. But let me be the patient. So what are you telling me, doctor? Should I take the probiotic or not?
Go ahead. Yeah.
Okay. So you got the answer, right? Well, go ahead. I will give the answer of Seth, right?
But just to go back to, I think Seth raises a really interesting point, right? The microbiome study is one of the things that we have. It's something new. It's something novel. We need people to participate in that. We had a meeting yesterday between the Montana group and between our group at NYU, which was really exciting. And the data is going to change clinical practice. It's going to change our understanding about the disease. A lot is going to come from that. We're always talking about patients that have chronic diarrhea, right? We have within the FD community a number of people that have diarrhea that we cannot control. We're trying, so there's an unmet need there to discover treatments. Particularly if you have a GI problem, if you're one of the patients that does have chronic diarrhea, we need your samples, right? What we have in FD are not too many people, so we need extremes. We need the extremes of people that have a lot of GI problems and the extremes of people that don't, because somewhere by comparing and contrasting those two groups, we're going to be able to have more answers about the microbiome. So particularly if you have problems with the GI tract, or if not, give us those samples, reach out to Maria, Dr. Kotrina at our center who is coordinating that, because we really want to push that project forward because we have a lot of hopes for it.
And there are also, look, I don't want to steal the thunder because I hope they present next year. There are some incredible findings, even not just the microbiome, but all these metabolomics. It seems to be a big absorption issue. Right? There seems to be maybe a mitochondrial energy utilization issues. There are a number of questions that we have that their initial results are being, perhaps that's why I'm saying would be a game changer. We are learning a lot of things from that study and things that can be operative, not just the probiotics. We may be able to do more, right? So yes.
Boring question. Is there a correlation, or do we know at this point if there's a correlation between gallbladder disease and FD? Is there a higher number population-wise?
Yeah, the answer is yes. There seems to be, we are not sure. Look, let me answer easy. Usually, the gallbladder in medicine, there are these things that medical students remember. It's usually females. They are obese and they're very young, right? The people that have. That's not the case in FD. Clearly, there is another problem, and we did analyze some of those things, and there seems to be a motility problem with the gallbladder. Normally the gallbladder has to contract, and that requires sensory and motor innervation. So many patients with FD, more than what you would expect in a similar sample, have that sludge, that lack of emptying, and then stones. So how fast you decide to operate or not operate, a good number of patients got the gallbladder removed. So the answer is yes, the incidence is higher, and we believe it's a neurogenic problem because of the contraction. Yes?
The PTC 258, the thought process on that then, is that to help slow down the nerve degeneration and just the overall frequency and speed of the complications, but it's not a cure per se for the genetic disposition, but rather just to help slow things down?
Correct. Well, you are correct, and then it is a cure, quote unquote, in the sense that it corrects the splicing defect and it results in the production of normal amounts of the protein that in FD is reduced or absent. So that's really all we know. We know that we are correcting the defect and making the protein normal again, normalizing the protein. How that will modify the disease, that's what we do not know for sure. We know indirectly a number of things that are very exciting. We know, Sue showed us that, and her team, that mice that have no protein at all don't survive. They're not even born. If you give them a little bit of the normal protein, you make them survive. And now, as you show in the mice, because for the first time we can also do that, there is a correction of the number of neurons in the DRG, which is that acronym, that's the dorsal root ganglia, and one of the consequences of lack of nerve activity, which is your ability to control the muscles, that results in the bones in the spine not developing correctly. That they also fix. How exactly that happened, how you go from that protein to the bone, the eye, and the nerve, that's the $100 million question. We would like to answer that. We still don't know that. So we are looking for things that will improve, that we predict that will improve. Now, we can only find what will improve if we know exactly what worsens. You cannot fix what ain't broken, right? And if it's broken completely, or if it's not there, you cannot fix it either. So it's a difficult dance. You have to find something that is progressively worse, but is still there. If the neurons don't develop, you cannot fix them. So you need to find the neurons. For example, we think that the gait, the balance, gets progressively worse. And you tell us that. You tell us that they have more trouble walking. Well, we think that more trouble walking is because the surviving neurons of one type, something is wrong with them and they probably decay. So we think that maybe this protein will make those surviving neurons not die. Now, we measure exactly in detail how the coordination, the ability to walk, how that worsens. Well, it worsens, but when we show that to the statisticians of PTC, they say, yes, you show us 100 people, a lot of variation, and yes, everybody got worse. Now, you need to show us one guy in 6 months, how much that worsens, and how you can measure it if we improve that by 20%. Well, Eisenson, you tell me how to do that. I'm trying to do that, right? So those are the challenges we have. We know things get worse. We know things get worse. We need some way to modify that. Yes. Hands up. I thought it was Lucy calling me on the phone saying time's up. That's George.
I'm sure we all have a lot more questions, and I thank the panel for all the information that you provided today, and invite you to stay on for the schmooze afterwards, and hopefully some of you can ask your questions face to face with our panelists.